All right, hey, it's day 4. First of all, I just want to apologize for the really awful editing on yesterday's video. So my introduction got cut off and then I got cut off at the end. But anyway, hopefully we're kind of going easy on each other during this time. So this is day 4. And yesterday we talked about DNA and we compared it to language. So you remember we talked about the fact that we have the bases in the language of DNA, and a base in DNA is like a letter. And then the bases combined together, 3 bases make up a codon, and that's really important to remember that it's 3 bases that make up a codon. So there's 3 bases in a codon, and the codon is like a word in DNA. These words combine together in DNA, and words combine to make, or codons combine to make genes, and that's like words combining to make a sentence. And then the genes make a particular protein. So these genes are going to make a particular protein. That's one of the things we're going to be looking at today. And then the genes group together to make the chromosome. And that's kind of like the chapter of a book. And then the last thing that we said yesterday is that you take the complete set of chromosomes in an individual, and that's called a genome. Okay? And that's kind of like the book. Okay, so this is a really important idea to understand, how we've got this language of DNA in which everything that your cells need to know to make you are in the language of DNA. Okay, we talked about that yesterday too, so you should know that already. Make sure that you have that down. You also should remember that there are 4 different bases in the language of DNA. You do need to remember that they're adenine, thymine, guanine, and cytosine, but the most important thing to remember is that adenine always pairs with thymine and guanine always pairs with cytosine. Those are really the most important things to remember. Yesterday, where I got cut off on the video, I was talking about the fact that what happens with DNA now is that it unzips. So in its normal state, you've got the twisted ladder. Who can remember what the twisted ladder is called? Yes, the double helix. That was probably Avery that answered that question. So the double helix, or the twisted ladder of DNA, looks like this. Like that, though, the DNA really isn't good for much. If those individual base pairs, or codons, simply stay within the DNA strand, they don't do anything. They just sit there. So this is sort of information locked away in the DNA. In order for that information to be useful, the DNA has got to unzip. So the DNA, with the aid of specialized proteins, whose job it is to help unzip the DNA actually then unzips like this. So you'll have it kind of unzipping, exposing those individual bases like this. Okay, so those bases are exposed. Now, we'll put the letters on for the bases. So let's just say down here, this was A, G, C, C, G, A, T, 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 G, C, G, A. Okay, let's just say that that's the sequence that we've got. And by the way, you'll notice that there's a picture on the website from Jurassic Park when the Velociraptor is kind of going through the laboratory, and the the characters in the movie are hiding up in the ceiling grates, and the velociraptor sticks his head up in the ceiling grate to see if he can see the people. And you see the reflection of the lights coming down from the ceiling grate. And at first glance, it looks like they're just squares on the velociraptor's skin. But if you look really close at that picture, you'll notice that they're not squares. What the movie producer has done is made all these little tiny letters, A, T, C, G, C, G, A, A, T, T, G, G, C, all these little genetic sequence letters reflected on the dinosaur to sort of be a hidden message about the nature of the film. Anyway, let's just say that this is our sequence of DNA, and those, of course, would be matched by their complementary bases up in the top. What happens is that another protein comes along and helps facilitate the matching up of a new strand of DNA. So we get a new sequencing there, like this. So see, here's this codon. It was AGC. It's being matched on the other side by a new group of nucleic acids that are TCG. When this whole matching takes place, we get a new strand of DNA. And so this new strand of DNA would twirl up again in its double helix pattern and be a duplicate of the original. When would this happen? Well, this would happen when a cell divides and becomes two different cells. And that sort of process is happening all the time. We're going to be talking more about that process next week. But that process is known as mitosis. So when we talked about scraping our cheek cells, and I said that your cheek cells are constantly producing new cells, that's the process of mitosis. Body cells are produced through this constant dividing of one cell into two cells, and then the new cell dividing into two new cells as old cells are sloughed off or die in the body. It was interesting because yesterday I was just reading that from when you're 4 years old until when you're about 50 years old, nearly all of the cells of your body, except the cells in your brain, have been replaced. I think that's kind of cool. That's kind of a cool thing. So it's this constant process of replacing cells, and that process is called mitosis. But one of the things that has to happen in mitosis is the DNA has got to replicate itself. So this process where you've got DNA making more DNA is called replication. Now, this process is similar to another process that has to do with how DNA actually works. So you might remember in that video that we watched about viruses, that in order for the instructions that the DNA carries to get out into the cell where it's actually going to be used by the ribosomes to make the proteins, it's got to be carried out into the cell. The DNA doesn't leave the nucleus. The DNA stays put in the nucleus. The instructions have got to go out into the cytoplasm of the cell to the ribosomes where the work actually gets done. In order for that to happen, we need to understand another molecule. And that other molecule is called ribonucleic acid. Now, you'll notice that ribonucleic acid is really similar to deoxyribonucleic acid, but what it doesn't have is the two different rails of DNA. It doesn't have that D or 2 prefix in front of it. It's just ribonucleic acid. Ribonucleic acid is otherwise known as RNA. And a really important type of RNA is called messenger RNA. It's got a little lowercase m in front of it, mRNA or messenger RNA. And that mRNA is created using this same unzipping process as creates a replication of DNA, but instead of this becoming a new strand of DNA and this up here becoming a new strand of DNA, like what would happen in replication, this section right here would stay a single stranded strand of ribose and would unzip again from the code of DNA that it replicated here and travel out into the cytoplasm of the cell to bring this gene to a protein where the protein would actually start the process of making the protein. That process is called transcription. Again, transcription is where the information goes from DNA to messenger RNA so that the information from the master plan in the nucleus can get out into the ribosomes in the cell. And that's where we're going to stop today. So hopefully you've gotten your reading assignment done. Your reading assignment should be due tomorrow. Hopefully you've also watched all the videos that are also added to this posting, and you have given me a call, or you've sent me a text, or you've sent me an email if you have any questions. I look forward to correcting all of your work in your Google Docs, and we'll see you next time. This has been Day 4. See you guys.